So I'm going to start by giving some background perspectives with some of the feedback and input that I received from you. Some of the needs are to have a community-based system by which we can curate and annotate sequence and genotype data also across databases. And for that, we will need to have agreed-on standards, and primarily with the germplasm as the entry point for connecting the genotypes and sequences to phenotype data. We also need, I think, to have updated phenotype databases where ontologies really serve as the framework for the curation of that data. And beyond that, we all know that we need to develop new tools that will facilitate smart breeding, accessing germplasm and the sequence information to proceed into alleles and sources of germplasm that can be used in, for example, marker-assisted breeding. We also need to ensure that the stocks that are being sequenced and genotyped are publicly accessible. For example, the purified lines from gene bank accessions that have gone into the sequencing effort. So one aspect, one way of potentially looking at this is that we have different databases handling different types of data, germplasm pedigrees, genotype sequences, phenotypes, and then the tools for breeding and for annotation, for visualization, and for query cut across those different types. At IRIC, we have the International Rice Information System, which is the data store that the gene bank, passport, and other information is stored in, as well as many pedigrees for IRIC lines, as well as other lines in the public domain. This is just one example of a database that could be connected to such a system. At this time, we're all very familiar that we're experiencing data overload. There are many now, thousands of genomes that have been resequenced from specific genetic stocks. One example is the 3K project that you've heard about between CAS, BGI, and IRIC. And there are many other projects. One example is Ben Han's activity in China. Another example, the 120 genomes that have been sequenced for the SNP discovery pool for the Rice SNP Consortium. Another project going on in Japan is the resequencing of their core collection. We also have now thousands of purified genetic stocks that are being made accessible for phenotype. So ultimately, we're going to have many phenotypes at multiple locations for these stocks. So how do we manage and handle this state? As well as the potential of generating different types of omic data sets from those same stocks. The 3K project initiated by CBCAS, BGI, Shenzhen, and IRIC, and it's the first phase of an initiative to potentially sequence 10,000 lines. And ultimately, we would like to be able to plan on sequencing all of the gene bank stocks in our collection, as well as those in our sister centers under the Global Rice Science Partnership, and then extend that if it seems a feasible thing to do. In terms of sequencing data that's been obtained now under the 3K project, we have a little over 3,000 genotypes that are now in analysis with a minimum depth of 4x and an average depth of now between 13 and 16x depth. Some of those go much higher. And at this time, the data is being uploaded into GigaDB. That's the database associated with Giga Science Journal, which is a collaboration between BGI and Biomed Central. in terms of some initial analysis of that sequencing data, the SNP diversity, the SNP polymorphism extending across the genome. This shows a summary for about 20 million of those SNPs that were identified by an early analysis. There were also a number of indels detected across the different types. This is a summary of that in about 50,000 of the indels discovered. An initial analysis of that also illustrated that we were able to capture the diversity within rice. All 5 variety group types were covered by the sample that was used for sequencing. So this is just to give you a flavor of what's going on in the 3K project. At the start of the discussion this afternoon, we could go into more detail regarding that. Summarizing some of the input and feedback from various people, there are numerous other activities that we're all aware of in rice. Blake Meyer's team, for example, with small microRNAs, the small RNAs, and associated analyses. Blake is involved in the International Arabidopsis Information Consortium, and he sees that as one point, IRIC should coordinate the activities for an informatics portal for rice. It needs to have a specific type of mechanism in place for self-governance, answering to stakeholders and other partners. It should not, it should build upon other model systems. It needs to adopt and adapt as much as possible, and it needs to be an open access type system, and it can serve potentially as a mechanism to attract funding across international borders, and the germplasm should be easily accessible and in the public domain. Input from Detlef Weigel. They have immense resources at Max Planck Institute Tübingen for computation, and of course you're aware of their involvement in the Arabidopsis 1000 genome project, where they now have on the order of 500 genomes accessible in the public domain for that. Our partners at IRD and CIROT have a number of activities for deep sequencing and the use of genotyping by sequencing across different lines, and then that's feeding into different phenotyping activities on materials for trait and discovery. They also have several of their own database systems, Origins, Greenfield DB, TropGenes DB, and a database of tag lines, mutant collection of tag lines that are resources and tools for rice genomics. They see that having a common portal where different information systems can be accessed is one of the key components that we should strive for. They also see that it's necessary that we integrate across these different sources of information, and that crucial raw data like NGS data be in a common repository and with a standard type of format. And apart from that, it's also necessary that we have a framework for regular meetings and ways that we can work as a community for applying for funding.
Ken, we just got an email from participants. They were asking if they're not getting video transmitted out. Are you trying to transmit? Are people signed in for the video on this? No video.
No video.
Okay.
And GRAPPA has bioinformatics laboratory, and they are equipped with hardware structure for supporting NGS and other types of data analysis. Wageningen plant breeding, they have they're currently resequencing in tomato and brassica. They're set up for other types of sequencing projects, and they're wanting to make use of sequence diversity and breeding applications. Richard Pinker sees that some of the issues that we need to address, how we will handle storage of big data sets. How can we integrate data sets to accomplish meaningful statistics such as genome-wide association studies? How do we integrate across different data stores? And one approach for that would be to make extensive use of semantic web ontology. His team is working on tools for visualization of multiple genomes and complex genomes, and also ways to link function to different annotations for breeding application. And then we're all aware of Ramin and the tremendous effort put forth for comparative genome analysis, including much of the rice data being in Ramin from OMAP and from other efforts, and there are many other data sets curated in Ramin. Transcriptomics, microRNAs, SNP data, et cetera, and there's a comparative view across the ERISA genus. And then Pankaj Jaiswal and others, the team is working on developing a reference ontology for plants that would encompass not just the plant ontology, but trait ontology and environment ontology. And those ontologies are going to be critical to connecting, to being able to accurately and precisely annotate phenotypes in a way that will make them, will curate them and in a manner so that they can be cross-queryable. And these ontologies are already being set up in a web services framework that will allow that to be done, as long as the databases that are holding such types of data make use of the ontologies. So just a very brief summary of input. One framework that could be used as an example for the path forward is the International Arabidopsis Informatics Consortium. And as much as possible, we shouldn't be, we should build on existing efforts and tools. The genetic stocks and germplasm really need to be the entry point for this effort, so that the data is truly relevant for the application, which is in breeding. And having a rice information portal that would allow this to happen might be the key mechanism by which it can be done. Also, we need a community agreement on standards for the way data is annotated, the way data is curated. We need to use the ontologies, different IDs like a germplasm ID that's shared and agreed on as a way to access information about a specific stock of a germplasm across different data sources. And that could be done through semantic web or web service enabled technology. We need to have tools that will integrate the big data sets. And we need further development for visualization and analysis tools to access the information. That's just an attempt to summarize information that I received from partners. If there are gaps in this, you can blame me, I hope in the discussion that those gaps are filled. And with that, I will turn over to Hakeem Dover.